By Montserrat. As already nicely introduced by Montserrat, my name is Denise Mitrano, and I'm Assistant Professor of Environmental Chemistry of Anthropogenic Materials at ETH Zurich. I've been here for the last few years, but I've been studying this topic of microplastics and nanoplastics in the environment for several years now. And we've been using some new analytical tools from my previous work in engineered nanomaterial analysis and trying to apply these tools to understand this new suite of contaminants. So when we're thinking about anthropogenic activities and the environment overall, and the impacts on the environment, we all know there's a lot of different stressors that are facing the environment today. So there are obvious big things like climate change, biodiversity loss, deforestation. But in our group, we're mainly focused on anthropogenic particles. So anthropogenic particles can be things like soot emissions, engineered nanomaterials, or the focus of our research now and the focus of this talk, microplastics and nanoplastics. So when we think about measuring these particles in the environment, first we need to be able to develop appropriate tools to be able to detect them, quantify them, and characterize them in terms of shape, size, and their chemistry. We also then, once we have these tools in place, to assess the microplastic inputs and their implications in the environments. And as environmental scientists, I don't think we only want to describe the problem, but be also able to suggest some different solutions to these aspects. So our work also tries to support regulations and eventually design and develop more environmentally conscious materials so we have less negative impacts of plastic pollution in the future. Now, as Montserrat mentioned, we have a lot of topics about plastics in the news today, but the first overarching theme that I'd like to underline is that plastics are not all the same. This is really one overarching umbrella that contains a lot of different types of polymers, plastic added chemicals, and different compounds, which are in the different types of products that we use every day. And these different types of polymers and plastic added chemicals can all have different effects in the environment and on biological systems. And so keeping in mind that when we say plastics, this can actually be a really heterogeneous group of compounds. But it's not only the compounds themselves that we want to be able to look at. There's also this size continuum of plastic pollution, which is quite important. So a lot of plastics that get into the environment are these macroplastic waste, bottles and plastic bags, which get into the environment. And through UV or mechanical stress, they can break down into smaller and smaller sizes. And this is called microplastic, those things that are 5 millimeters and below. Now, at this size scale, we can have different biological impacts, such as easier uptake by organisms, or aggregation with different natural particles, which will eventually change their fate and transport in different types of systems. Now, this degradation of macroplastic to microplastic doesn't stop there. We can also form even smaller particles called nanoplastic. This is normally things that are 1 micron and below. And these, again, can have different types of interactions in the environment because of these smaller sizes. So we might have increased transport in water or soil systems or in the atmosphere as well, and also different interactions with organisms, potentially even cellular uptake. Another important thing to consider when we're looking at anything that's made out of a particle is the surface to volume ratio, where more surface is available and interacting with the environment. And so we might have a leaching of additives that is more common in smaller particles as opposed to larger ones. So with this whole idea of that size really matters in terms of fate and hazard and ultimately risk of plastic in the environment, you might be asking yourself, so we have smaller plastics, does that mean that we have bigger problems? And I'm sure everybody has seen these type of headlines in the popular media that there is some beautiful place, but it's poisoned by microplastic. Microplastic is found in the snow from the Alps to the Arctic, so it's everywhere that we've taken a sample. Microplastics found in our tap water, and are we even eating our fleece jackets because there's microplastic in our food? Okay, so I give these examples as a little bit of hyperbole and a lot of clickbait about what the public might think about microplastic. But what do we really know about this topic as scientists that are working in the field? So what is really known and what is really unknown in this area of microplastic and nanoplastic research? So even though many tests have been studied to date, there's still little eco and human toxicological effects that are certain. So there's a lot of uncertainty here about if it is the polymer chemistry, which is important, the particle size, or the plastic added chemicals, for example. While plastics have been measured every place on Earth that has been studied so far, essentially, there's still really little information on the dominant fate and transport processes that are delivering these particles around the globe. And in that sense, in both of terms hazard and exposure, the risk assessment is still subject to a lot of uncertainty. But what we can say with more certainty is that microplastics and plastics in general are a very persistent material, which is widely dispersed across the environment and with many unknown ecological consequences. And so it's certainly something that we should start to investigate further. So how can we start to do that? First, we need to be able to have some tools which are specifically able to measure particles and particles that are polymers. So what are the challenges of detecting anthropogenic particles in the environment? Whether you take a water sample or, of course, a soil sample, it's not only the particle of interest that's there, there's also a lot of other particles that you need to distinguish between. And this can be more challenging compared to other types of contaminants, which you might be interested in studying. So especially when we consider the differences between analyzing soluble chemicals and particles, we really have to have a change of mindset because we have to look at a lot of particle-specific parameters. That's because particles compared to soluble chemicals can have different fate, transport, and biological interactions. There's different variables for the risk assessment because of this, and therefore we need to develop new methods that are specifically targeted at particle analysis. So there's a lot of challenges with particle analysis in general, not only just plastics. We need to differentiate the particle types. So is it this actual anthropogenic particle, which we have in mind, or is it just a natural particle that's there? We oftentimes have very low concentrations. They're very small size, so we need to have very sensitive methods. And these particles can also change over time, either in their surface chemistry or their size, which will also be wanting to be investigated. There's a lot of additional challenges of detecting nano and microplastics, even though there's a wide variety of different types of particles that are in the environment coming from many different sources, as I show in this kind of puzzle piece of where all the different types of plastic pollution can come from. A lot of this comes down to analytical challenges. So the current analytical workflow is currently very time consuming. This is because if you don't only have to go out and collect your sample, but you often have to extract the microplastics from the sample, purify it, and then do some detailed analysis, which can take quite a long time. Further complicating this is that there's currently no standardization of either the methods or the materials to be able to use. And so therefore, it's really hard to know when you've gotten the right answer once you get an answer. And a lot of the techniques which are currently most common for microplastic analysis, such as micro-FTIR or micro-Raman, have comparatively high detection limits in terms of particle size, as well as concentration. And so that will make it especially hard to measure these smallest size particles, nanoplastics, using the current analytical technologies. This has a few implications about what we can find out about microplastics and nanoplastics. Firstly, because the analytical workflow is time consuming, we can only take a limited number of samples in the field or in the laboratory. And this gives us not a very good temporal resolution about what is happening, but we also can't test everywhere geographically to see where the particles are. Even in the laboratory, then, this analytical workflow makes mechanistic studies very time consuming. And as a consequence, we have a really reduced understanding of these basic processes of how particles behave in the environment. And so in this way, it's really difficult to make a risk assessment of plastics, especially microplastics and nanoplastics in the environment now, because we have to clear a lot of these analytical bottlenecks to be able to more easily measure them. So we, in our lab in the last few years, have tried to develop a little different type of approach that we could at least use in laboratory-based systems to more easily be able to measure nanoplastics and microplastics in complex media. Specifically, we have made metal dope plastics as this alternative approach, where we can synthesize own plastics with a metal dopant inside, put it into a number of different test systems, which I can detail later, and then analyze this metal as a proxy for plastic. Now, this is really helpful because there's a lot of standardized analysis for trace metals analysis. And so then we can more easily detect the fate and transport of microplastics and nanoplastics in our test system, as well as the uptake into biological organisms. So any technique which is suitable for measuring metals is now suitable for measuring plastics, which is great. So oftentimes we use things like ICP-MS, but also other types of synchrotron-based techniques, which are also suitable for metals analysis. So to just give an example of some of the types of materials that we make, we can have nanoplastics, which are made out of polystyrene or polyacrylonitrile, polyester fibers, similar to those that are in your textiles and clothes, and also fragments, microplastic fragments of different types, PLA, PET, and different size fractions. So here, the materials that we make are only model materials. So we tried to hope that they're environmentally relevant through some different aging and weathering processes, but still they are only model materials. But we have a very low metal content, which is in these particles, about 0.3% by weight. So it doesn't change the material chemistry very much in terms of density. And all the metal is on the inside of the plastic. So this important aspect of surface chemistry is retained about how it would be as a real microplastic. Furthermore, we've also assessed this metal doesn't leach over time, regardless of the test system that we put it in. So it's really a conservative tracer to be able to measure our plastic particles in a number of different test systems. We find that this approach has several different improvements. It's much faster. Instead of only analyzing a few samples a day by micro-Raman or micro-FTIR, we're able to totally digest samples and measure by their metal content to measure hundreds of samples a day by ICP-MS, for example. It's much more precise, and we can measure smaller sizes of particles as well, including nanoplastics. And most importantly, we can test many experimental parameters under controlled conditions in the laboratory because we can have a much higher throughput. And in this way, process studies can become much more possible to be able to understand fate, transport, and biological uptake of nanoplastics and microplastics. So now that we have this nice tool in place, how are we actually using this to assess plastic inputs and implications in the environment? So we have a lot of ongoing work in this space. So if we think about the environment as a whole, a lot of plastic pollution comes from urban centers because these, of course, have a lot of anthropogenic activity. So we have tested the retention efficiency of microplastics and nanoplastics in wastewater treatment plant to see how much goes to the sewage sludge versus how much is released in the wastewater stream. We look at how this is transported down rivers and eventually into larger water bodies, the effects of aggregation with particles, and then consequently their settling dynamics. And also the sorption of organics like natural organic matter and metals onto the plastics. While I'm not an ecotoxicologist by training, I work with a lot of ecotoxicologists, so we can also look at the uptake and impacts to a wide variety of different organisms. Since we know that plastics are in our water sources, these water sources could also be used as drinking water. So we have also tested the suitability of current drinking water treatment chains for water purification to remove nanoplastics and microplastics. When we think about not just aquatic systems, but also terrestrial systems, these can also be impacted by microplastics. So from this wastewater treatment plant in 50% of European countries, this sludge, which contains most of the microplastic and nanoplastics which enter the wastewater treatment plant, can be put onto agricultural fields as a nutrient amendment. So then we can also look at how microplastics are being run off from these different agricultural fields, how they're transported deeper through the soil column, how their uptake into different plants, and also how the incorporation of microplastic into the soil system can impact soil porosity and greenhouse gas emissions. We also work with some colleagues that are ecotoxicologists that are more soil-based, and so we can also look at the bioturbation and uptake of microplastics from soil-dwelling organisms as well. And so this is, of course, a wide range of different types of work that we do in this space. But for the context of today's presentation, I only wanted to highlight one of these types of studies to kind of show this utility of these metal-doped plastics. And also, I think, could be interesting to kind of myth bust some of these microplastics in drinking water. So we'll look a little bit closer on how drinking water treatment plants are able to purify water for public consumption. So why do we want to look at this removal from drinking water treatment plants? We know that microplastic and nanoplastic are already in many different drinking water sources. And so our question was, can conventional drinking water treatment plants already remove them? Or do we need to have some sort of upgrade to drinking water treatment plants to have better water? And specifically, which different stages of the drinking water treatment and the operational conditions of the drinking water treatment plant are most efficient at microplastic and nanoplastic removal. So maybe not everybody is super familiar with what exactly is happening in a drinking water treatment plant. So to give a small overview of how the drinking water treatment plant looks, at least in Zurich, the raw water for drinking water is taken from Lake Zurich. It then goes through a pre-ozonation condition to kill a lot of the bacteria, a rapid sand filtration, activated carbon filtration, slow sand filtration, and then finally it's distributed into households. And a number of these different systems could be responsible for removing particulate matter, not just microplastics, but all particulate matter from the drinking water treatment chain. And this especially is the rapid sand filtration, the activated carbon filtration, and the slow sand filtration. So we wanted to investigate these different processes in different stages. So we had both laboratory column experiments to be able to assess the removal under these different conditions, pilot scale drinking water treatment plant conditions, and then finally taking these results to model how a complete drinking water treatment chain would work and if it would be effective for nanoplastic removal. So when we look at the laboratory scale column studies, this was great because it gave us flexibility to study nanoplastic removal on a small scale system. And so in concept, what we're trying to do here is take our metal doped plastics, nanoplastics in this case, put it up through this column and then see how much nanoplastics are coming out the other end by taking this water, doing microwave acid digestion and measuring by total ICP-MS to again see what is the retention through these different columns. We were able to do a systematic evaluation of the different water treatment changes by filling what is in the column with different substrates. So this could be sand or activated carbon, which either contained a biofilm or not, which is typical in a drinking water treatment plant. And we can also look at other important parameters for nanoplastic removal, such as flow rates and other operational conditions of this test system. So of course, when we had a longer column, we were able to remove more plastic by simple filtration. And of course, a lower flow rate also removed more nanoplastic. But key here is that we also found that the sand column was removing more nanoplastic than the activated carbon, especially when it was covered in a biofilm. So here we found that the slow sand filtration system, which is the one that has having most biofilm associated with it, would be most effective at removing nanoplastics from the system. So these laboratory scale systems are great, but we also wanted to see if this worked on a larger scale. So to have this increasingly realistic, we looked at a pilot scale drinking water treatment plant in Zurich. And this was done with the water supply company of Zurich. So we took really the raw lake water, which is coming into the plant, had a nanoplastic injection before it was put through these two large filters. One containing the sand with a biofilm on top, naturally occurring, and the other one with the activated carbon, which also contains some biofilm. As you can see from the photo, we had several different sampling ports through the depth of this column, so we were able to get a resolution of where the nanoplastics were being retained throughout column depth. And we could also take solid samples of the substrate, either the sand or activated carbon, at 4 different depths throughout the column study. And so here we could start to make these breakthrough curves of nanoplastics in the pilot scale studies. And I can appreciate that maybe on your screen, this is a little bit small to see, but I can kind of walk you through these graphs in a little bit more detail. So our total exposure in the drinking water treatment plant was for 6 hours that we were putting nanoplastics through this system. And then we stopped adding nanoplastics so that we could see how long it would take to move through the entire column. On the y-axis, we have the concentration that was measured in that certain column height with divided by the total amount of plastic that we had put into the system. And we can see through the different heights that more plastic was retained in the upper layers of the activated carbon filtration system compared to the lower layers of the system. So the upper layers were responsible for most of the removal. When we look at the solid content, so actually taking the activated carbon, we're able to also see this similar trend that more nanoplastic was captured at the beginning of the treatment, and further through the column depth, there was less nanoplastic that was there. We saw quite a large difference compared to the sand filtration, where we saw a much higher retention of nanoplastics in this slow sand filtration column, where we're only getting 20% breakthrough in these first few centimeters, and basically complete retention further down through the column. And this is also supported when we look at the actual sand substrate, that we're only measuring nanoplastics associated with this really top layer where they were mostly captured. So using this approach, we had a possibility to measure nanoplastic retention over the filter depth. And this was in agreement with our lab studies, where we had a higher retention in sand than an activated carbon. And this was specifically due to this additional biofilm layer, which was in the top of this sand filtration unit. Now, when we take these breakthrough curves and start to model them for having a simulated performance in a complete drinking water treatment chain, we can start to see the impacts of sequential removal across the treatment chain. So we had a high cumulative removal efficiency, even at quite high nanoplastic concentrations, much higher than environmentally relevant concentrations, where we had a 3-log removal across the entire treatment chain. And the slow sand filter was the most important removal step that was tested, and where we had 99% removal through the sand filter. However, what's important to note here is that there were no breakthrough there was no breakthrough modeled until about 3 months of continuous use at this high nanoplastic concentration. But typically we would also have a backwash cycle to regenerate the filter, so we wouldn't anticipate that much nanoplastic that actually goes through the drinking water treatment chain. So in sum, we can see that we tested the drinking water treatment chain, and it significantly reduces the nanoplastics in potable water. And we could maybe even anticipate high removal in a full-scale system, because they have additional treatment steps which weren't modeled here, such as a coagulation step to remove the large particles. And there's even a more established biofilm there than in our pilot scale systems, which we found to be the most important factor for the nanoplastic removal. However, it's important to say that these results can't necessarily be applied to all drinking water treatment plants everywhere, because we only tested one type of nanoplastics, this polystyrene nanoplastics, and so different polymers may behave differently. And also drinking water treatment chain configurations can also be behaving differently. So this slow sand filtration step, for example, is now often not being included in a lot of new drinking water treatment plants because it takes up a lot of space physically, and it's also more expensive to operate. And so it's also quite important to be able to see how this is going to affect different types of drinking water treatment plants in the future. So how can these type of results start to support regulation in this space? So in a few recent reports, either from the World Health Organization or SAPIA, they've specifically highlighted what is the situation with microplastics in drinking water. And basically both reports had said there's not enough information to really definitively say one way or another if there's actually risk. More studies are needed. So some more studies are now being done. So for example, at the California State Water Board, they've just put a plan in place for the next 4 years to monitor microplastics in 700 different drinking water treatment plants across the state of California to be able to start to understand what is the real exposure to at least the folks in California, and if they can start to correlate which different types of drinking water treatment plants are most effective at microplastic removal. However, even in these studies, because they are limited by the current analytical capabilities with micro-Raman and micro-FTIR, they're not able to measure particles that are below about 20 microns in size. And so it will miss a lot of potentially smaller size particles that could be in the water supply, but they would not necessarily know. So what are the considerations on how we can have less plastic pollution in the future in general? This is always a trade-off when we think about how we can use plastic in the future. So we can think about which type of feedstock selection that we actually make new plastics from. Do we have petrochemical based sources, which we know the oil and gas industry is a pretty dirty industry overall, or should we rather use bio-based plastics? Now, bio-based plastics can be good when we think of using food waste, for example, but there could be some perverse incentives of actually using food sources to make plastics. So again, the feedstock selection can be considered. We can have better material optimization. So do we want biodegradable plastics or those that are more persistent? Biodegradability is often considered or said in the public media that this is the solution to plastic pollution, but biodegradability itself is really dependent on where the plastic ends up, and it doesn't necessarily mean that a biodegradable plastic, which is suitable to biodegrade in a compost, would actually biodegrade when it's in the ocean. And finally, we can look at the product design and use, single use versus long-lasting material. No matter what the product is and what material it's made out of, single use is oftentimes not the most sustainable alternative. Underlining all of this is to have a better material and chemical simplicity so that we could start to move more towards a circular economy for plastics to have easier and more feasible recycling. And of course, having some economic incentives, because plastics are so used now because they are so inexpensive. But if we can find some other economic incentives to offset this price, then that maybe different materials would be chosen in the future. And so that is my little tour of anthropogenic particles in the environment, from developing analytical tools to assessing their implications. And with that, I'd like to thank you again for your attention and be happy to answer any questions.